Good day. I would now like to proceed with the second part of section 1 of the beef cattle breeding limited online lectures. What I would like to do in this part is to talk about the two different ways of inheritance, if you could call it that, of genes. We've now established that genes are basically the code on the chromosome that carries or causes differences in appearance or performance of animals. So I would like to divide this type of inheritance into two different types. The one we can call Mendelian inheritance, named after Gregor Mendel, a famous geneticist, and also then the second part, so-called quantitative inheritance or polygenes. So the first one is what we also could call simple inheritance, where only one or a few genes are involved with causing the differences in measurements or observations in animals, whereas in the second instance, many genes, each with a small contribution, would cause the difference or the variation in animals. So we will then kick off with simple inheritance or Mendelian inheritance to start off with. Like I said, it's a simple inheritance. It's not influenced by the environment. So what you observe in animals would be purely because of gene actions, and not there won't be differences in the expression or the observation due to possible differences in environmental influences. We also see dominance effects. You can recall from the first lecture, there are different forms of the gene. One would cause, let's say, one coat color red, and the other one will cause another coat color, let's say black. They are located on the same place or spot on the chromosome, but the one will then influence the other, or the one can dominate the other one. So these are then the examples: red and black coat color, for example, or polar horned animals. Let's take the example of coat color, where we know that the black coat color, or the gene causing black coats are dominant over red. So you could have a red animal will always have what we call small a, that's the gene for red, because if the dominant black gene would have been present in this animal, it would have had a black coat color. You'll remember that chromosome comes in pairs, so the genes come in pairs. You inherit each of those from your sire and dad. So a black animal, if an animal has a black coat appearance, it could either be because it's carrying both sets of the black gene, or it can carry the black gene and the red gene. But because the black gene is dominant over the red gene, it will not be possible to observe or to distinguish between a carrier and one that will breed pure black animals. So you can have an instance where you have a pure black animal being mated to a pure black animal. The bull has got both. In both counts, he has the dominant black, and a female will have the dominant black, and we expect that in all cases, all progeny, all calves will have a black coat. What if we mate a red bull to a red cow? A red bull can only be red if it has what we call the recessive sample of the gene, and that would be red, and a red cow. So in all cases, all progeny will then be red coated. What if we mate a black bull that's pure black to a red cow obviously she can only have the recessive gene for the coat color, which is red, what will happen to the progeny? The progeny will all be black because black is dominant over red, but you will see that each and every one of these skulls, 100% of these skulls will be carriers of the red gene. They are carriers of the red gene, but it won't express as red because black is dominant over red. What if I made a carrier bull, that's a bull with the dominant black, but he also has the recessive red gene. If I made that bull to a red cow, what can we expect, or what would the probabilities be in terms of coat color? This is what we will get. We will get, in this case, we will get there's a 50% chance to 25% chances of a calf being a carrier of the red gene but not expressing it because the black gene will dominate the red gene. The other half or the other 50% chance would be pure red animals in the herd. So if you have a herd of red cows, let's say 30 red cows, and you made them to a carrier black bull, you can expect that half the calves would be red calves and the other halves would also be carriers, like the sire. What happens if you have a black cow and you mate her to, or black cows, let's say 30 black cows, and you mate them to a black bull, but he's a carrier black bull, so he's not pure black, he's a carrier of the red gene. So you will get half the calves would be pure black, that will have only have the black gene, and the other half will be carriers of the red gene, but all of them will be black coated animals. I think an interesting one is if you mate one carrier to another carrier. So in this case you have two black animals, they are both carriers of the red gene, and in this case let's say you have the 30 cows again, but they are all carriers, and you mate them to a carrier bull, you can expect only a quarter of those calves to be red coated animals. The half of them will be carriers and the other quarter will be pure black, or they will have only the dominant genes for that. So this is basically how this mode of inheritance works. In this case, so in this case you will have 3/4 black, 1/4 red, but of those 3/4, only 1/4 will be pure black animal. This is what we sometimes get in nature, where you have, like for instance, the lions, where the dominant color would be the golden color of lions, but you every now and again, when two carriers are mated, you can have a white line popping up because that's a recessive coat color. Another instance with Mendelian inheritance is we get modifying genes. So you can have the black or the red, but sometimes there's a dilution factor. Sometimes on another space or place on the chromosome that will influence the coat color gene elsewhere. Also spots and other color variants. A very important trait or property in terms of this type of inheritance is the pole gene. The pole gene is dominant over horned animals, so polled animals will have at least one copy of the dominant gene, the pole gene, but you will have animals that will breed pure, where they have both copies, they receive both copies from their parents, whereas in certain cases they will not breed pure in terms of the progeny. So how would you know whether a bull has will breed pure polled, or will only be a carrier of the polled genes. Because remember, in terms of physical evidence before you, both bulls will be polled because of the domination of the polled gene. So if you have a polled gene, one way of testing is to breed him to horned cows. So if you have a polled bull and you breed him to horned cows, let's say 30 cows, because a horned cow will not carry the polled gene. So if you have any horned progeny, it means that he was not breeding pure poll. So in a case where all the progeny is polled, he will breed pure. He will that would be then he will carry both. In both cases, he will carry polled gene, and if some progeny has horns, it means that he also has a recessive gene for horns. This is what also happens in terms of genetic defects. Usually they are recessive. You don't see them as often, but as a result of inbreeding, it means it increases the chance of both parents being carriers of the recessive unwanted gene. There are obviously laboratory tests and genomic tests available where you can pick up these or the possibility of carriers, but usually with the result of inbreeding. Inbreeding brings out those recessive genes because it increases the possibility of both parents being carriers of the recessive gene. One very interesting case is the so-called myostatin gene, that is being coined as double muscling. And you see you get it in different species, even in dogs and rats, and in certain cases, in certain production systems in Europe and so forth, it's seen as a positive. In any extensive production system for beef cattle, it's a no-go. It's an unwanted gene, and farmers like in South Africa would not want that gene to be part of their production system. This is a very good instance where we have a dam with a calf, and obviously she's a carrier because the bull was a carrier, the dam was a carrier, and it showed in the calf. So the calf basically then have both the two recessive genes in him, and it shows in the so-called double muscling in that. This is an example of a lab results. There are different types of variants of the myostatin gene, and you can see that there's an animal carrying both variants of the Q204 variant. There's another one, he's a carrier. This one is obviously, you can see it in the animal, it's visible in the animal, and in this case the animal only carries one sample of this, and it's not visible on the animal, but he is a carrier. The other zeros means that they are not carriers, or they have no copies of the variant. Just a few other side notes on inbreeding. We know that an increase in inbreeding is associated with the so-called fitness traits, survivability of animals, the mothering ability of the animals. It can also negatively affect growth and reproduction. It also leads to lower conception rates, more abortions, especially where you have these recessives that's hiding somewhere in the population, and it only comes out once where you have to carry, you make two carriers, and it can even cause prenatal deaths or embryonic deaths. So you can end up with more stillborn and weak calves, and also a higher susceptibility to diseases. So the rule of thumb is not to mate related animals. And another rule of thumb is to try and keep it below 6%, or at least the rate of inbreeding below 1% per generation. This is just an example of inbreeding. The pink blocks here would be the females in a pedigree tree like this, and the blue ones males. So in this case, you see a very low inbreeding percentage of 0.43%. At least for the two previous generations, there were no relationships amongst the animals. You will see there are some high inbreeding coefficients, 19.42% in this case, for this bull. So obviously he has parents in his pedigree that were mated, that were closely related. Another example is half-brother, half-sister matings. You can see here you have a bull that we used they are related. So in this case, it's a sire of a grandsire and of the dam and the sire, and you end up with an inbreeding coefficient of 12.5% in the progeny, which is basically double what should be allowed in terms of inbreeding coefficient. Sire-daughter matings, you can see you end up with a 25% inbreeding, the same sire, basically of the dam and this specific calf. You get very bad types of inbreeding. This female ended up to be 43% inbred. Same sire, generation after generation on the females. So this is things that can happen, but the chances are very big that you end up with animals with low immune systems or weak immune systems that will not survive, and that will be extremely lowly fertile. And this is just a service from Stadbook side. It's possible for breeders to do so-called mock matings to see where the whole pedigree tree is available for a breed, and you can log onto a system at Stadbook to check what type of inbreeding you can expect of progeny for certain matings. It's also then these schematic representations that you've just seen are examples of that's then presented to breeders and that they can use in their decision making. This is the example of what you get if you run a mock mating, and it will predict the expected inbreeding of a specific calf. If you, for example, in this case, this breeder said, what will happen if I use this bull on that cow, and will give you an estimate of or the calculation of the expected inbreeding of the progeny of that specific mating. That then brings us to the end of part 2 of section 1 of the beef cattle or the limited online lectures for beef cattle. We will talk again and talk about breeding in beef cattle in part 3.